Chapter 12. Chapter 12 is all about viruses. By the end of this unit, I hope that you can compare and contrast viruses and understand how they're different than bacterial cells and human cells or eukaryotic cells. I also hope that you can explain why viruses are difficult to treat with drug therapy and compare and contrast how viruses replicate based on their genome. Our opening case study involves a 25-year-old nurse who was dealing with a patient when she had an accidental needle stick. It turned out that the patient had hepatitis C virus. 6 weeks after this needle stick, the woman developed a fever and some flu-like illnesses, and because of her exposure, they immediately tested her for liver function and hepatitis C viral load. It turns out she was infected with hepatitis C virus. This is a virus that infects the liver cells and causes liver damage. She was immediately put on 2 different viral drugs to treat her symptoms, and while they reduced the viral load and improved her liver function, the side effects were very, very, very terrible. The drugs that attack a virus attack the human body as well, because viruses live inside of host cells. They don't live autonomously. They must live inside of a host cell. So to attack a virus with a drug, you have to attack the host cell that it lives in. So the side effects were really terrible. So even though she did have a viral count and her liver enzymes weren't perfect, they took her off of those medications. She seemed to bounce back and feel pretty good, but 10 years later, she then had another episode where she was feverish and had flu-like symptoms, and it was found that her liver function was deteriorating and she had a higher viral load again. They had to find new viral drugs because the virus had mutated within her body. This is an RNA virus, which is very susceptible to mutation. And so they had to find 2 new drugs. The new drugs on the market were incredibly expensive, and unfortunately this nurse's insurance would not pay for those drugs. So she was put on the liver transplant list. Now this is a really important infection to be aware of. Not many people talk about hepatitis C virus, but it is the leading bloodborne pathogen in the United States. 3% of the world is infected with hepatitis C virus. And while for about 15% of the patients this virus clears on its own, meaning you just sort of get better, there's 85% of the patients have chronic liver problems, chronic liver disease, and it can lead to liver cancer. It's the leading cause of liver transplants in this country. So this chapter is obviously about viruses, and hepatitis C virus is a great example of an RNA virus here. So that's why we're talking about it in this opening case study. But this case also points out why antiviral treatments are very, very difficult. Okay, so when we think about viruses, we need to first recognize that they are very, very small. So here's our scale bar of 200 nanometers, and you can see how tiny this RNA virus is. This is obviously taken with an electron microscope as well. As we tour through the virus land, we need to be aware that viruses have a very specific and definite host range. So while the hepatitis virus that we just talked about infects liver cells, it does not infect any other cells of the body. And hepatitis C virus that infects humans only infects humans. So it only infects humans. This virus here is called a bacteriophage, and this bacteriophage in particular infects E. coli cells. So bacteriophage are viruses that infect bacteria. When we think about the diversity of viruses, we can see viruses infecting every form of life on this planet. So we're just going to go over a couple different examples. So viruses that infect bacteria live inside of bacterial cells and then lyse or burst out of those cells, leaving a hole. If you grow a lawn of bacteria, you can see this opaque growth of bacteria, and you sprinkle virus on top, you can see where the virus has killed or lysed the bacterial cells in this example. Here is a virus that's budding out of a membrane. So you see the viruses are leaving the membrane, taking some of the membrane with them. So here we have a virus that's wrapped in what we call an envelope, right? And this particular virus is one that causes measles. So just like our opening case study, we know that there are a number of different viruses that can infect humans. And the viruses that infect humans infect different tissue types, so they have different tropism, whereas hepatitis virus only infects liver cells, measles only infects the skin cells. Here is a tobacco mosaic virus, and you can see the mosaic virus particles are these long, straight filaments here inside this cell, and this particular cell is a plant cell. So viruses can infect plants as well. Here is the outcome of that infection. This is a tobacco leaf that should be nice and dark green, and instead it has this mottled light green coloration. So viruses have a very specific host range, which means the type of organism that it can infect, and within a host it has very specific tropism, or it can only infect certain tissue types inside that host. When we think about viruses and we think about their host range, we also can appreciate the diversity in both shape and genome of viruses. So when we think about viruses, we classify them according to these Baltimore groups. So viruses can have a double-stranded DNA genome. So this means they have two complementary strands. In virus language, we call the different strands positive and negative. So if we have a positive strand and a negative strand, that's a double-stranded virus. And those would be complementary, meaning that if A is on one strand, T would be on the other strand. Some examples of those double-stranded DNA viruses are here. Group 2 are single-stranded DNA viruses. So most human and bacterial genomes are double-stranded DNA. Here we see some genomes that we've not seen yet. So a single strand of positive sense DNA is our group 2 viruses. Group 3 viruses are double-stranded RNA. So that's another category that we don't see in our cells. In the language of viruses, positive RNA acts as messenger RNA. So another way to say that is the ribosomes sit on positive RNA. Positive RNA is the complement to negative RNA. So if A is on one, U is on the other. Group 4 is our positive RNA. So that we can have single-stranded RNA. This is very similar to the messenger RNA that we would already see in cells. So positive RNA is something the cell is somewhat familiar with. This is what the ribosome sits on. We have viruses that come in as a negative sense single-stranded RNA virus. So they only have the negative or complementary strand to the messenger RNA. We have groups of viruses that start out as an RNA virus but then create DNA from their RNA. Now this is the opposite of what happens in bacterial cells and human cells, where we see DNA going to RNA and going to protein. Here we have RNA going back to DNA. So these viruses are called retroviruses because they go back, and they have a special enzyme that does reverse transcription. So it's reverse transcribing viruses. And then our last category, group 7, these viruses include hepatitis B virus, but otherwise a lot of plant viruses. And these are DNA viruses that go back and forth between a positive RNA virus and a double-stranded DNA virus. They have two different forms of the genome. Okay, so those are different kinds of virus genomes we can have. Now, as you can imagine, when we think about the structure of a virus, we have to appreciate that a virus is simply one of these genome types that is wrapped around protein, and that protein we call a capsid protein. The capsid protein is just used as protection around this viral genome. And that's one of the reasons that we have so many troubles treating viruses with antiviral drugs. So to clarify that further, right? So 3 reasons why viral infections are difficult to treat. 1, they're hard to discover. If you just have a genome wrapped in protein, there's not a lot of drug targets there. So in bacterial cells, there is cell walls and cell membranes, and there's ribosomes, and there's other types of enzymes that do metabolism. A virus has none of those things. So a virus simply has a genome that is wrapped in protein. So it's hard to find something to actually direct the drug to attack. Secondly, because viruses rely on a host cell, they live inside of a host cell and use all of the host cell's machinery, they have terrible side effects. When you try to stop the function of a virus, you have to stop the function of its host cell, meaning that you have to damage the human cells if we're talking about a human virus. And then second, because these genomes have such variety and the genomes are not normally found in the host cell that they infect, we often see a high mutation rate. So double-stranded DNA viruses actually have the lowest mutation rate, and that's because we have the polymerases that do this are really good at being accurate and checking for errors. But the polymerases that make these other viruses don't necessarily have that evolutionary pressure to preserve and keep the integrity of the genome. So we see lots of mutations, and that makes them difficult because if the virus has changed or mutated, it may not be susceptible to the same types of treatments and drugs that you've used before. Okay, let's talk about some of the structural types of virus. This is an icosahedral virus. So here you can see the capsid proteins are in blue and green here. So these are the proteins that are arranged in an icosahedral shape. So these kind of look like a nice symmetrical ball or an icosahedron with 12 faces on them. Inside, if we kind of look inside, you can imagine this red stuff in here would be the genome of this particular virus. another look at that, right? So if we look at a cross section, we can see the capsid proteins arranged along the outside and the DNA inside. So this is the basic form of a virus, a genome that's wrapped in protein. In this case, it's a DNA genome. And our virus capsid is arranged in an icosahedral shape. So that's our first shape. Here you can see a capsid again, right? And then we can have an envelope shape. So we have an envelope, right, that is oftentimes stolen from the cell membrane. So we see enveloped viruses. We can see filamentous viruses. So here you can see the genome is sort of arranged in a helical fashion, and the capsid is wrapped around the outside. Now this is just a small piece, but when we looked at that tobacco mosaic virus, you can see they turn out to be these long strands. One example of a filamentous virus like this looks like a long thread that's been twisted on itself, and it looks like in that example is Ebola. So long tubes are called filamentous viruses. Here's an example of that. You see these long filamentous strands. So you think like a bacterial rod, but really, really long. And inside that, the DNA is inside that long tube, usually in a helical formation. Okay, so we have this complex shape, and this one is really cool looking. Bacteriophage often, but not always, often come in this particular shape. So this shape has a head that is the genome. It contains the genome, and it's wrapped in its capsid. It's usually an icosahedral-like shape, but then it has sort of a neck and tail fibers. So this is all made out of protein. This particular part of the protein sticks and binds to the bacterial cell, and then the genome actually gets squirted through this tail into the bacterial cell. So this complex shape, I call it alien robot, right, is the type of virus that infects bacteria. And here you can see an EM, this is an actual electron microscope picture of this bacteriophage infecting the bacteria. Okay, and then lastly here again is a virus that doesn't necessarily have a rigid capsid, so it's a polymorphous shape. It can kind of get squished around, and often because of that protective envelope, it doesn't need to have as rigid of a capsid structure. Here's an example of what that looks like. So we have our genome, we have the capsid or the core proteins, and then we have the envelope. It's important to note that many enveloped viruses, so this is the envelope is typically made out of lipids, again often stolen from the host cell membrane, but in those lipids we have these spike proteins. Now spike proteins become very important because that is the protein, that's the point of contact between these viruses, these envelope viruses, and their host cell. So it's important to note that spikes oftentimes are targets of vaccines or of drugs because this is where the virus actually interacts and infects the host cell. Okay, so viruses, obviously, like I said, come as a genome that's wrapped in protein. They can sometimes have an envelope, and they can sometimes have some more complicated, complex structures like the bacteriophages. And they also have a lot of complexity in their genomes. So you can see that the viral genomes can be very simple and short, or they can be very complex. As we go through and look at how viruses replicate based on their genome, we'll see some more examples of how these viral genomes are arranged. Now, in the world of things other than cells, we obviously have viruses, which we've been talking about so far in this chapter, but there are two other types of organisms that we should pay attention to. One are viroids. These are simpler and more basic than even a virus. So a viroid is an infectious particle that is just made out of RNA. Right? Let that sink in for a second. So this is RNA that folds back on itself and base pairs, right? So we have a 5' end over here and a 3' end over here. And this viroid can act and cause disease and can cause changes in the host cell and can cause infection. But it is just a piece of RNA. Wow. On the flip side, we have prions. Prions are just protein, so they have no genetic component. They have no genome. And this is a infectious particle, it's a protein, that causes other proteins to change their shape. Prions, so far, we've only found one example of a prion protein, and it is found in the central nervous system. So this is a protein that's normally found in the cells of our central nervous system, in our glia, and it normally has this shape right here, and it's really important for transmembrane function. When the abnormal conformation, so when the prion protein is folded in a different way, Okay, this is the tertiary structure of the protein is different. The primary and secondary structure is the same, but the tertiary structure is different. When this abnormal form of the prion encounters the normal form, it causes it to shift into the abnormal version. Right? So this is an infectious protein. It has no genetic component. But the outcomes of this are really important because this abnormal conformation is not functional in the cell. And so the cell keeps trying to make more of this prion protein. But as soon as it makes the normal form, the abnormal form makes it to switch, changes its shape, creates the abnormal form. So this broken, diseased prion protein keeps building up in the central nervous system and causes cell death. And as you can imagine, we know that the central nervous system is really important. And so the diseases that these prions cause are devastating neurological diseases. Some examples are Creutzfeldt-Jakob disease, which is similar to a disease found in cows we call mad cow disease, or bovine spongiform encephalopathy. There's also a disease in humans called Kuru that's caused by these infectious proteins. Okay. So how do these viruses actually replicate? So here we see some of the diversity we've seen in the genome. We know that each virus has a different kind, each type of virus has a different kind of genome. And we know that the central dogma says that there's DNA, right, that gets made into RNA, that then gets made into protein. But if we think about how all of these different virus genomes work, they all have a slightly different path. So a double-stranded RNA virus will simply use the ribosomes to make messenger RNA. Right, so that we've already seen that. If we come in as a negative strand DNA virus, we'll make the double-stranded virus, which makes the messenger RNA. If we come in as a double-stranded RNA virus, one of those strands acts as the messenger RNA. And again, if we come in as a positive strand virus, that is going to act as the messenger RNA. Interestingly, if we come in as a negative strand virus, negative RNA virus, we have to make the messenger RNA in order to make protein. So it's important to sort of understand the relationship that each of these viruses has a slightly different job that it does to help make its proteins in the cell. Here is a general overview of the stages of viral replication, right? So this is the example of a bacteriophage. So here we have a bacterial cell. It has a host genome, and here comes our alien robot bacteriophage. It's going to attach to the host cell. And in this case, because it's a bacteriophage, it's going to inject in its DNA. So bacteriophages, the protein never gets into the bacterial cell. That's very different than in human viruses, as you'll see in a minute. So after attachment, the genome comes in and does biosynthesis. So the genome helps direct and make new virus proteins and new virus genome. So the virus, if it comes in as a protein with the genome, it needs to leave as a protein with the genome. And so both of those jobs need to happen during biosynthesis. So the bacterial cell makes the virus proteins, makes more of the genome, and then the virus particles get assembled in the assembly step and maturation. Once the virus particles are complete, the cell gets lysed, the bacterial cell dies, and the viruses are released. Now, interestingly, some viruses, in addition to having this type of lytic cycle where it simply is a virus factory and makes new virus, right? Every virus does this pathway here on the left, right? But some viruses can actually integrate their genome into the host cell and hide. Now, the example we're seeing here is in bacteriophage, but we'll see some examples of animal viruses that can do this as well. So once the virus genome gets into the host cell, it can just simply incorporate into the genome. So see here how the virus genome is in dark purple, and it just stays there. And it can stay there and stay there and stay there until the virus just keeps multiplying alongside the bacterial cell. In human cells, this happens the same way. So the virus inserts its DNA into the human chromosome, and every time the human cell divides, the virus genome gets divided as well. At some point, your immune system is dysfunctional and can't keep track of these viruses, or there's some other stressful incident that occurs, and the virus comes out of hiding, pops out its genome, and goes back into this lytic cycle. So again, every virus has this lytic cycle where it goes through and it makes new virus particles and it kills off the host cell. Some viruses can go through and do the lysogenic cycle where its genome hides in the host cell's genome and does not make any protein. So over here, you do not see any protein or virus particles being made. It's just the genome that gets copied. Okay. How do we grow viruses? So we know that viruses must grow inside of a host cell. So we can't just sprinkle viruses in a broth and expect them to grow. They need all of the elements of their host cell. So if we're trying to grow a bacteriophage, we would take some bacteriophage, and we would mix it with E. coli, right? So in that mixture, we would have both the virus and the host cells that the virus requires. We would pour that on top of an agar plate, right? And then we would see these plaques or holes in the agar where the virus has grown. Here's an example of what that looks like. So we see the bacteria, and each of these little dark spots is where the bacteria has been killed, and we call those plaques. They're kind of the opposite of a bacterial colony. How do we grow animal viruses? So animal viruses are trickier to grow, but basically what we do is we have to grow the cell that the host cell that the virus uses in a dish, right? So if we can't grow that type of cell in the lab, we can't grow the virus that infects it. So that can limit a lot of the virus research that we can do. But we can grow these cells. Sometimes these are cell lines, like the HeLa cell line. Sometimes they are tissues that we simply take out of an animal and can grow for a short period of time in media. But basically we grow the animal cell that the virus infects, right? And we add the bacteria on the viruses on top, and then we start to look for these plaques or areas where the cells have died. So here's an example of that. So here is areas where the animal cells have lysed or died or been eaten away by the virus. So the key thing about growing viruses in the lab is you have to be able to grow their host cell. It's kind of like if you had a lizard, you have to also have crickets, right? So you have an aquarium of crickets and aquarium for the lizard because you have to have both, right? Okay, so let's go into some specific examples by using some case studies. So our first case study to think about animal viruses is a 19-year-old college student. He's a member of a fraternity and had a lot of fun with all of his fraternity brothers and with various women at parties. And he'd gone to his campus health center because he'd had a small lump on his penis, and he was worried about it. He knew that it was abnormal. Well, sure enough, the nurse informed him that it was likely that it was human papillomavirus that had caused a genital wart. The freezing of the warts is the only effective treatment at that point. And you can imagine that's not very fun on your sensitive genital areas. So freezing the warts is a way to treat this infection, but prevention is really important at this point as well. So the nurse advised this young man to make sure to wear a condom and make sure to inform his partners about the risk. But one of the things that we have to be careful of is this virus infects skin cells, so epithelial cells, keratinocytes. And so a condom tends to not to cover all of the skin that comes in contact during sex. So you have to be very careful and be very aware of the potential to have and spread this disease. Now, this disease can be spread through both vaginal sex and anal sex, as well as oral sex. So it can infect the anus, it can infect the genital area, and it can infect the esophageal cells in your throat. And it's been known and linked to different kinds of cancer because this particular virus, we can see here that it is icosahedral shaped, right? It is a double-stranded DNA virus. This particular virus goes into lysogeny. It does that trick that we just talked about where the virus genome gets incorporated into the skin genome and stays there. Now this virus, because it is relatively benign, meaning it's just a small lump on your genitals, it doesn't seem all that serious, it's easily transmitted and not taken as seriously as it should. 80% of adults over the age of 29 have this virus in the United States. 80%, right? Because this virus is linked to different types of cancer, there's been a vaccine. Now, this particular young man who's 19 and already been exposed to this virus cannot benefit from this vaccine. The vaccine must be completed before somebody becomes sexually active, before there's even a chance of somebody becoming sexually active. And so this vaccine becomes a little bit controversial because we give it to very young children, and it is a sexually transmitted infection. Okay, so why is this particular virus a great example for us to talk about? Well, this is a double-stranded DNA virus, and it infects the keratinocytes. So you can see here it infects the skin cells, and then it causes them to grow abnormally, which turns out to make these warts, but it also can transform those cells and cause cancer. Here is the genome of the human papillomavirus, and I want to point this out because it's kind of fascinating. We know that the reading frame of DNA, right? So we divide up our DNA or our RNA into 3 bases, which make a codon. But we know if we shift those over, we would get totally different amino acids. Well, viruses have capitalized on that, and they have figured out a way to make 3 times as many proteins from one genome. So you can see here this genome. If you read it in one reading frame, you get this protein and this protein. If you read it in a second reading frame, you get this protein, and this protein. And if you read it in the third reading frame, you get this other protein out here. So that's pretty interesting and efficient of these particular viruses. Here's our little icosahedral human papillomavirus image from an electron microscope. Now, like I said, this is a DNA virus, so let's talk about what it has to do. It has to bind to the host cell, and again, this is going to be one of those receptors that's found only on skin cells, epithelial cells. It's going to bind and get brought in through an endocytosis, right? Once it gets in, it has to then get into the nucleus. So it's going to dock into the membrane. It's going to get its genome out. So it's going to take off all of its proteins. This is a double-stranded DNA. So it's going to get copied by just the regular DNA polymerase that's in the cell. So we see more copies of the genome that get made. It's going to use the ribosomes and the RNA polymerase to make messenger RNA. Right? So here in the cytoplasm, we'll see it getting made into protein. So the RNA gets made into protein just exactly like any other RNA in the cell would get made into protein. But those proteins, those capsid proteins, will come back into the nucleus to help assemble the virus particles. Those virus particles will build up into the cell, and the cell will lyse out and release more of that human papillomavirus. Okay. Our next example is from an 18-year-old freshman in college. Her campus was experiencing a flu outbreak, and they had told all of the students that if they felt ill to call a special number on campus to make sure that they could be put in isolation and not spread. Well, sure enough, our patient got a fever, was put into isolation, was given food and drugs, and made a full recovery. So that's a great story, but we know that 10% of the United States are infected with this particular virus, the influenza virus, every year. Most severely affected are young children and the elderly. So this is a really important pathogen, and one that we'll talk more about when we get to vaccines. This is an RNA virus. So it's important to remember that this is a negative strand RNA virus. And interestingly, influenza has put its virus into 8 chromosomes or segments, right? And because of that, these segments oftentimes get switched out. So we hear the reporting of flu viruses like H1N1, and that's because they have very different features to them, and that's because this virus is easy to mutate because one of these can get swapped out with any other chromosome and create a slightly different virus. So here we see the chromosome. The chromosomes are segments of this virus genome. This is a negative strand RNA genome. We can see the capsid proteins, right? So these yellow capsid proteins here, and we can see that this virus is enveloped. So we have a phospholipid bilayer, and we have these spike proteins. These spike proteins come in 2 flavors, H and N, so this red one and this sort of tannish one, and that's where we number the types of viruses that we see, so H1N1, right? Okay. Here is an example of how the viral diversity happens in influenza. Now, influenza has a host range that includes avians, like birds and ducks and chickens, pigs, and humans. So it can infect 3 different hosts into the respiratory epithelial center. And because of that, there's a chance that this particular pig might be infected with a bird virus and a human virus. And that's where we start to see these segments start to recombine, and we get new and different flu viruses that emerge. Here is our RNA virus and how it replicates. So this RNA virus first has to attach to the right receptor on the cell, okay, get brought into the cell through lysosome fusion. It then uncoats, takes off all of its protein capsids, and releases the negative strand. The negative strand travels into the nucleus, and positive strand gets made, right? It also gets capped, and some of that positive strand that gets made gets put into ribosomes. So we see the ribosomes here translating the 5' messenger RNA into proteins, right? So we see some of those proteins getting made. Some of the proteins are going to turn into spikes, and so they have to go to the membrane. So you'll see some of the proteins that get put into the endoplasmic reticulum and the Golgi. And some of the proteins travel back through here to get assembled, right? So packaging proteins return back to the nucleus. So some of the negative strand virus, like I said, gets made into positive strand, which gets made back into negative strand. Unfortunately, the way nucleic acids work is when you copy them, you can't just make a straight copy. So you can't just put an A across from an A. If you have an A, you have to put a U. And so if we make a copy of this negative strand, it'll end up being positive strand. So this particular virus has to make positive strand as a template to make more negative strand virus. So that virus genome gets made. Like I said, those proteins all got made. Everything gets put together. The envelope happens with the capsid spike proteins, and the virus comes out here. Each one of these steps is blocked by a different kind of drug. And one of the most effective drugs we have here is Tamiflu, and it can block the virus from exiting the cell. And if it can't exit the cell, it can't go off and infect new cells and make new virus. Okay. Our last example is a 24-year-old car salesman named Ralph. He had been with his current girlfriend for a couple of years and had not been unfaithful to her, and he was getting ready to get serious and propose to her. He thought first he should probably get a full physical exam that included sexually transmitted disease testing. So he got tested, and it turned out that he was positive for human immunodeficiency virus, HIV. He had no symptoms of this virus, so he was very, very shocked. And through a number of different counseling sessions, he was then prepared to go back and talk to his longtime girlfriend. Now, why is this virus infection so serious, and why did he not know that he had it? He had it. So let's look at this. So immediately upon infection, we can see the viral load, which is this red line right here, go up. So right after getting infected with HIV, the virus immediately spiked. And during this period of time, people experience a mild flu. So they might feel very, very sick and run down, maybe even have a slight fever. But we call this sort of subclinical, meaning most people don't go to the doctor for it. And unless you've been engaging in very risky behaviors that you divulge to your physician, you're not going to get tested for HIV at this point. So they call this acute HIV syndrome. Now, this particular virus infects T cells, which is a very important cell in our immune system. So you can see that immediately upon infection, those T cells get infected and die. Now, notice that there's a break. So once we immediately have this virus spike, it goes right back down again. And that's because this virus, just like human papillomavirus, goes into latency. It can hide in our genome. That period of time can then be anywhere from 1 to 11 years. It's a very, very long period of time that cannot really be predicted. And then once it comes out of that, we sort of start to see symptoms and opportunistic diseases occur because the T cell count begins to drop slowly, the virus load begins to increase, and at some point the immune system is affected by the loss of these T cells, and people are susceptible to diseases. At this point, we then talk about it as clinical AIDS, or acquired immunodeficiency syndrome. Here's a picture of this particular virus. This is an RNA virus that goes back to DNA. So we call this a retrovirus. So here is the structure of this. So we see these RNA viruses. Notice that unlike some of the other viruses that we've talked about, this virus packages some enzymes. It packages a special protease, and it packages an enzyme that takes its RNA and makes DNA with it. So it's called reverse transcriptase. It also packages integrase. Now, inside this genome, there's a capsid, or outside the genome is a capsid, and an envelope with some spike proteins. It has a genome that's relatively simple and includes all of the enzymes that it needs, as well as the enzymes for its envelope and its polymerase and its copying itself. Okay, so one thing I want to point out on this replication diagram is that in red are all of the different antiretroviral drugs that we have and the steps that they block. So we've made a lot of progress in preventing HIV infection from becoming a fatal disease, and if managed properly, it just becomes a chronic illness rather than a death sentence that it used to be not all that long ago. So here we have this RNA virus attaching to the cell. It uses some specific receptors that are found on T cells and occasionally some other types of monocytes. The virus gets in, that RNA genome then gets made into DNA using that enzyme reverse transcriptase. That DNA then incorporates into the host genome. This is where it can go into latency and simply stop doing anything else. At some point, though, the virus will enter into the lytic cycle and start to make RNA, messenger RNAs. Some of those messenger RNAs get made into proteins that are capsids. Some get made into the other enzymes that it needs. More RNA is made and transcribed. Some of the spike proteins that are used are made here in the cytoplasm and used to make the envelope. And so here we see that this virus can then bud out and leave after a certain period of time. Interestingly, they also found that if two cells are touching using these CD4 or CCR receptors, the virus can actually move from one cell to the other within a nanotube, basically, without having to sort of go out and infect the other cell. So that's another mechanism that they know about, that HIV virus can spread within the body. Okay, so that is the end of our study of viruses, and we'll be learning more about it in our activities.